Look closely at this laboratory vial. Suspended in the clear fluid is a microscopic, pulsing cluster of cells. It has an intestinal tract, the early folds of a brain, and a beating heart. This is a living mouse embryo, but it was created without an egg, without a single sperm cell, and it has never spent a second inside a uterus. That was in 2022. By 2023, scientists successfully replicated this exact same process using human cells. By growing these organisms in a lab, researchers have rendered the traditional biological requirements for mammalian reproduction unnecessary. In a normal pregnancy, the moment a fertilized egg implants into the uterine wall, it enters a biological black box. We cannot directly observe how those early cells organize themselves inside the human body. Yet this hidden window is exactly when things go wrong. It is the period when thousands of pregnancies fail, and when developmental defects like spina bifida first take root. Scientists have long wanted to study this critical window in a petri dish, but they are blocked by the 14-day rule. This is a strict international ethical guideline that forbids growing actual human embryos in a lab beyond 2 weeks. Researchers at the Weizmann Institute of Science realized that if they couldn't legally study real human embryos, they would have to build accurate replicas. Bypassing the human body was the only way to cast light on the most fragile, unobservable stage of our own development. The idea of growing an organism outside the natural body is called ectogenesis. The term was coined a century ago, in 1924, by British scientist J.B.S. Haldane. It took nearly 100 years for a team led by biologist Jacob Hanna to make it a reality. His method eliminated the need for sperm and eggs. This relies on naive pluripotent stem cells, cellular blank slates that develop into any tissue. They don't need fertilization, just a chemical bath to activate their programming. Hanna's team separated these cells into 4 groups, instructing them to become the building blocks of an embryo. By swapping gametes for programmable stem cells, the researchers engineered a path around the normal cycle of human reproduction. When those 4 types of chemically altered cells were mixed back together, they began to move, fold, and lock together into a single complex structure. This self-assembling cluster generated its own yolk sac and the support cells required to build a placenta. It is known as a stem cell-derived embryo model, or SEM. The resulting synthetic model is nearly identical to a natural 14-day human embryo, mimicking its structural and morphological layout. If you find the bleeding edge of human biology as fascinating as we do, take a second to like and subscribe to the channel. The blueprint of human life contains all the instructions it needs to execute its own development inside a glass dish. The immediate goal is understanding early development, but long-term applications point toward functional artificial wombs that could assist patients with damaged uteri. Some feminist theorists have argued for decades that artificial wombs could eventually free women from the physical risks and biological burdens of pregnancy and childbirth. But an artificial womb would also shift the threshold of fetal viability. If an embryo can be grown outside the body, then viability technically begins at the moment of conception. This creates a paradox in abortion law. A patient could exercise their right to remove the fetus from their body, but the fetus itself could simply be transferred to an incubator and survive. By making pregnancy optional, ectogenesis renders the current legal frameworks of reproductive rights obsolete. And right now, these lab creations are operating under a legal catch regarding that strict 14-day rule. Because they are generated from stem cells, international guidelines classify them as models, not embryos. They exist in a regulatory vacuum, exempt from laws governing human embryos. But researchers are pushing to see what happens as these unregulated models surpass the 14-day limit. We are approaching a philosophical cliff. If a lab-grown organism looks, acts, and develops exactly like a human embryo, does its artificial origin actually matter? Science has figured out how to build the foundation of a human being from scratch, but society has yet to decide what to call it.